Right. Hello, everybody. I think we are live now, and I'm Joni Rutter. I'm the director of the National Center for Advancing Translational Sciences, and I am here today to kick us off. I won't be the main speaker today, but I did want to provide a few comments before we get started, and I kick it over to Hytham Sidki. Um, so, welcome to everyone who's joined today, and I know that some people are probably continuing to join. So while that's happening, I wanted to give you a few housekeeping items um, that I wanted to make sure everybody was aware of for this particular webinar. First of all, the forum is being recorded, and the recording and the slides that we have will be available in the coming weeks, probably about 10 days to 2 weeks from now, just to make sure that we can get everything in order and upload it to our site. Um, Also, for this particular webinar, all attendees will remain muted and will not have access to the chat. So with that, I think those were the main housekeeping items I wanted to go ahead and relay. But I also wanted to start off and kick off this sort of welcome for this webinar with an apology. I've talked to this community a couple of times or different groups within the community a couple times over the last 6 months or so. And our original plan was to hold this webinar in March, about 3 months ago. And so here we are now, 3 months later. There's been a lot that's really happened here at the NIH and at NCATS, and our team may look and feel a little different now, but we've been here the whole time. Everybody who you'll see today has been here the whole time. We've been working hard behind the scenes to continue building N3C into a world-class resource that it has been, and then it will continue to be for the community. So today we're really excited to not only sort of reconnect with the community, but also to share the broader vision that we have been working on. And the reason that we have taken a little bit longer to do that is because we have been trying to work out how we can build not just with the resources within N3C and make them better, but also to establish how we can really intersect and integrate with other resources around the NIH. And that's something that we hope to describe a little bit more in detail in the future. And so today, what we'll introduce is sort of the key folks on our side who are helping to carry the vision forward. And I think you'll see really how we've been able to build and grow N3C to a really wonderful resource that it can be for the community. I've sort of likened it to a metaphor that I've been thinking about. You're all probably familiar with the caterpillar and the butterfly, but N3C was born out of urgency. It was born out of this need to rapidly respond during a time of global crisis. But like a chrysalis, that what began as a very focused area of growth and an important foundation for what we have, this very time-sensitive effort has been transforming. And now we believe it's really ready to take flight into a butterfly. And our vision is essentially it's a powerful vision, I think, and an evolving resource that can go far beyond what we built it for and its original intent for COVID. But like any butterfly, it won't go far without the right environment, without the support. And that's really where you, as the community, come in. And we want to hear from you. We want to continue to get your insights now that we have built a stronger foundation internally. Now we can start to build that infrastructure and foundation externally as well so that we can be much more integrated overall. So to the entire N3C community, thank you. Thank you for your collaboration. Thank you for your commitment over the past 5 years. That's really helped shape a data platform that's already made an incredible impact on patient care and public health, especially through the acute and long COVID research that we've been able to move forward. We have over 5,000 citations. We have an H-index of 33 for N3C. We've attributed at a scale that's quite incredible in terms of collaborations with over 1,589 authors who have contributed to the N3C literature. We've transformed care guidelines in transplantation, in HIV, and in the use of Paxlovid for treating acute and long COVID. We've also generated evidence-based disease definitions over the course of this period, especially in the realm of long COVID, and we look forward to continuing to do those sorts of evidence-based disease definitions for other diseases as well. We've also developed complete risk prediction models for vaccines and for comorbidities related to the work that we've done. And I think that all of this work, again, is because of the incredible power of the community that we have, and our real interest and drive to working with the community to advance this even more. So today you're going to hear how N3C has expanded since its early days, where it's going next, and understand how we're committed to making it sustainable, secure, and a deeply valuable resource for tackling a wide range of health challenges. And again, your input today is going to be vital. So please ask questions. Share any feedback. You don't even have to ask questions, just make a comment. We're happy to hear those as well. We really want you to help guide what we do next, now that we're in this position to really kick this off moving forward. So thanks again for being here and for being part of what we hope will be the next great phase of N3C. Let's keep building this. Let's keep innovating and discovering insights together. And so with that, I want to turn it over to our lead for N3C, Haitham Sidki, and he's going to go through what we've really been focused on of late, and I hope you'll share the excitement with us. So Haitham, over to you.
All right, thank you, Joni. I'm going to go ahead and share my screen. All right. Hopefully everyone can see it.
Yes, we can see it.
Great, thank you. Yeah, so thank you, Joni. And what I really wanted to do was talk about N3C, talk about its past, its present, and its future. So, as Joni mentioned, N3C was born out of urgency. So the initial context for N3C was really an urgent need for observational data at scale. And, the context was, of course, the pandemic. And we all know here in the US, there's no centralized healthcare. And as a result, there's no centralized healthcare data. And even for one person, data for that individual can be spread across multiple providers, both in time and in space. And so N3C was born really to solve that problem. And then now kind of in the post-pandemic space, the question is why expand? And I think as we all saw through the numbers that Joni highlighted, COVID-19 was a very powerful proof of concept. It really demonstrated the value of large-scale clinical data harmonization and collaboration. And it showed that the significant investments and teamwork that we've all made have created a robust platform and governance model that can actually be expanded for broader use. And we all recognize that beyond COVID-19, there are chronic diseases and health disparities and other complex conditions that are still a critical public health priority. And so we would love to leverage and utilize the N3C infrastructure as a way to provide valuable opportunities as well for training researchers and clinicians and students in real-world data analysis, informatics, and collaborative science. And we've done a little bit of that, and we'd love to see more of that as well. And so the image here is supposed to kind of exemplify the initial collaborative COVID logo and show that it can kind of morph and expand into different areas like cancer and education, and renal, and all kinds of other disease and domains. So summarizing N3C, past and present, N3C has grown out of COVID into a national translational research infrastructure. And at its core, it combines harmonized EHR data, scalable governance, and team science to accelerate discovery across diseases and institutions. In the top right-hand corner, there's a QR code, and that's a QR code to the N3C Google Scholar page. So N3C was launched in 2020, in response to the pandemic. And 5 years later today, we have over 5,000 citations and a great H-index, and all the things that Joni highlighted in terms of how N3C has been transformational is very much due to the efforts of this community. Now, simultaneously with that, we've had 2 clinical pilots that have been ongoing. The first one was a phase 1 clinical pilot, and this was just to demonstrate the feasibility of expanding beyond COVID-19. We piloted clinical tenants for Alzheimer's, COPD, and renal disease, and there were 12 institutions from you all that participated in that effort. Right now, starting in 2024, last year, through to 2027, we've had the phase 2 clinical pilot. And that really built on the successes of phase 1. And the idea there was to scale with enhanced privacy-preserving record linkage, to focus on data integration, so to bring in CMS data and disease registry data like SEER and SRTR, and to support new tenants, so new disease areas like cancer and renal. And what we've seen both on kind of the COVID side and through the clinical side is continued engagement from all of you. So as context, there are 96 data contributors that signed the original COVID data transfer agreement. 76 institutions signed the COVID data transfer agreement extensions. So that provides researchers with access to institutions' data for another 5 years, so they can continue doing research on issues like long COVID and related conditions. We've had 12 institutions participate in the phase 1 clinical pilot, and right now we have 18 that are participating in the phase 2 clinical pilot. So thanks to all of you, we've had really continued community engagement throughout the lifespan of N3C. And so as a refresher for everyone here, some of you may be plugged into N3C. Some of you may have been more involved in the past, and you need kind of a refresher on how N3C works. And so what I'm hoping to do is kind of refresh everyone's memory so we're all on the same page, and we can talk about N3C today and in the future. So at a very high level, N3C takes in data from healthcare institutions across the country whose data are represented in different common data models, so different formats. These data are harmonized, ingested, and curated, and made available in the secure N3C data enclave. We leverage the OMOP common data model, and we leverage Spark, and all of this lives in the Foundry platform. And then through various access controls, collaborative analytics teams can get access to these data and carry out research and collaborate and produce all of the things that we just talked about. So that is how N3C functions at a high level. Now, kind of zooming in and looking at the data governance and access model, there are really 3 components that work in tandem to make N3C successful. There are the data contributing institutions. So data contributing institutions, they sign the data transfer agreements. They have IRBs in place, and they are the ones that produce the limited data set that gets contributed into N3C, into the platform. NCATS, we are the data stewards of these data that are being contributed by the institutions. And we bridge the gap between the institutions that are contributing data and the data users, which are the research community. So data users, their institutions enter into an agreement with NCATS, which is the data user agreement or data use agreement. They register, they abide by the community guiding principles, and then they submit data use requests, testifying to agreeing to follow the code of conduct and making sure that they carry out the necessary trainings, like IT security and human subjects protections training. And then they can request one of 3 levels of access to data in N3C. That's either synthetic data, which is computationally derived. It can be de-identified data, and then it can be a limited data set, which provides additional zip codes and dates of service. There's a data access committee that reviews all submitted data access requests. And depending on the requested level of data, there might be an additional requirement for a determination from an institutional IRB in order to access level 3 data. So kind of a brief overview, that is the structure of the N3C governance and data access model. Now, I wanted to talk a little bit about the phase 2 clinical pilot model, because this is going to serve as a foundation for some ongoing discussions. So the model I just highlighted is in place. So that shared governance model exists. So this is like a different slice. We're looking at a different aspect of N3C. And this relates to the clinical pilot model. So in this model, we are trying to scale to accommodate different disease areas and disease domains within the same centralized N3C framework. And so the way that works is data contributing institutions, they provide N3C with patients that meet what we call a disease agnostic phenotype. So that's patient inclusion criteria. And very roughly speaking, it's patients with 2 visits that are at least 30 days apart. And so these data enter into the N3C data enclave. This is a holding tank. It's locked down, and there's no research access to these data. These data enter into N3C. They are harmonized, they are curated, and they are linked via PPRO to registry data. And in one example would be commercial mortality data. And they're locked down inside of this holding tank. And then data contributing institutions have agency over participation in the different tenants. And so data flows from the holding tank into the different disease or domain-specific tenants using disease-specific phenotypes. So there are patient inclusion criteria for the renal tenant, for the cancer tenant that are specific to each one of those. And then the data contributing institution decides and says, hey, we want our data in cancer, or we want our data in renal, or we want our data in both. And then within each of these tenants, investigators can establish a number of research projects, which happens through the data use request process, which I outlined previously. Now, what we had initially envisioned building out this clinical pilot is a world where there's really a tenant ecosystem. So imagine the model here with cancer and renal scaled up, where there are now cancer tenants, renal tenants, education tenants, COVID tenants, diabetes tenants, pulmonary tenants, all kinds of different tenants that ultimately pull data through different phenotypes from this holding tank. And then we have investigators coming in and submitting projects within each of the tenants available here. So this was kind of the initial envision model for N3C on how to scale across new disease areas and new domains. And just to really highlight the objectives of what we were trying to accomplish, because we'll kind of come back to talking about the clinical model. With the N3C clinical pilots, we were trying to accomplish a few things. Number one, we were trying to understand both the financial burden, the infrastructure necessary, and the community resources we have to put in place in order to develop and maintain future tenants. So what we wanted to do was pilot these things at a small scale, understand the costs and operations that are necessary in order for us to be able to project that outwards and build something that is going to be sustainable for the community. And part of that is also refining the operations and the governance and also the technical architecture of N3C. It also gave us time to establish partnerships with CMS, HRSA, NCI, and NIDDK. And these partners have led various components of the clinical pilots. It also gave us time to work on next generation healthcare interoperability, and this is through HL7 FHIR US Core. And all of this together was supposed to facilitate new capabilities that will really expand the space of the scientific questions that can be asked within N3C and be answered as well. And throughout this entire process, we received feedback. Now, this feedback came from both the institutions participating in the pilots, from the broader community through a variety of different surveys or questionnaires that we sent out, and through a variety of meetings that we've hosted. And so these are just a few highlights of the kind of feedback we received, but we received a lot. I think institutions, they articulated the need for streamlined and robust agreement tracking. So oftentimes an institution, the institutional signing official might leave, and then that person get replaced, and then they need to locate their documents. And as it scales, there's a need to be able to have kind of a more streamlined and efficient way to track agreements, especially with the initial agreements being in place, and then the extensions, and then the clinical agreements. And so that can get quite complicated and tricky to manage. We've also heard feedback on there being more granular institutional user management. So, grad students, they graduate, postdocs move on to positions, people move around, and an institution having a good understanding of who is operating in M3C under their data use agreement is feedback that we've received because there is accountability in place there. We've also received feedback that institutions want to have more granular control of their data. So in the M3C clinical model, the institutions decide whether their data will be contributed at the tenant level, so in the renal tenant or cancer tenant, but some of you all communicated to us that you would like more granular control of your data, perhaps at a project level. We've also received feedback around access to institutional data. So within M3C, original data use requests, they're valid for 1 year and they are perpetually renewable. In order to facilitate research, once an investigator gets access to an institution's data in a data use request, they maintain that access in order to finish things like publications and make sure they're able to complete their research. But really, there were no time bounds around data use requests. And that was feedback we received from institutions that they would have hoped to have time limited, or at least like an understanding of the scope of time associated with each data use request. There's also been interest expressed in other disease areas beyond cancer and renal and the others that were part of the clinical pilots. Many of you all have researchers that are subject matter experts in other disease domains outside of the ones that we have been piloting. And then we've heard this, across the board, which is that investigators want to maximize the use of the linked data sets that M3C can provide on a national scale, and a lot more feedback. And so we really went back to the drawing board and we took all of that feedback to heart and we said, how can we incorporate this into the M3C of the moving forward? And so I wanted to cover one of two major changes coming to M3C. The first one is around data access requirements. So this aligns both with new requirements for what are called controlled access data repositories and feedback that we received from institutions around more granular user management. So now this is a bit of a heavy slide. It's filled with text, but we'll work through an example together, and there will be more information about this that will be sent out before this goes into place and becomes operational. At this point, just kind of, it'll be good to get an understanding of what's to come. So the first thing is M3C is now a controlled access data repository, CADR. And what that means is that there's a little bit of like a shift in terminology. So data use requests are going to be called data access requests. And then for every institution, there will be an institutional signing official, an ISO, that will manage an institution's users and their data access requests. And the other thing is that data access requests can be submitted by a requester who meets the following requirements. They are a permanent employee at their institution, so they hold a position equivalent to at least a tenure-track professor or senior researcher, and so that does not include a lab tech or a trainee or a postdoc or a grad student. And that data access requester should have oversight responsibility for all additional individuals named on a DAR who will be granted access to the data, and they'll ensure that all data usage fully aligns with the terms of the institution's data use agreement and the institutional policies that govern data access and use. And what that means is that individuals who don't meet these criteria, so whether they're trainees or grad students, they can still participate in any project, but they must be included under the supervision of an eligible data access requester. So how that operates kind of in practice. So here's an example of the CADR requirements in action. So on the top half of the slide, we can see we have a professor, Dr. Smith. We have a grad student, John Smith, and we have a postdoc, Jane Smith, no relation. And all three of them, they go through the M3C registration process. All three of those registrations go out to the institutional signing official for approval. And then once that approval comes through, all three of them will have access to the M3C enclave at large. And then when it comes to submitting a data access request, so for example, we want to study disease X in population Y, The professor, Dr. Smith, submits that data access request, and then John Smith and Jane Smith, the grad student and the postdoc, are attached to the data access request submission that Dr. Smith puts in. And then that submission goes through the institutional signing official for approval, and then that goes out to the N3C Data Access Committee for approval. So that, kind of in a nutshell, is how the CADR requirements will function. All right. So now what I wanted to cover is really the meat of this community forum, which is the next phase of N3C. So what I wanted to do was cover really each challenge that was faced and each piece of feedback that we received, and then how we have structured N3C in order to address these. And really the first thing that we wanted to touch on is how N3C can scale efficiently across clinical domains. And what we've learned from the clinical pilots is that really each new disease area requires a significant time and effort to design the cohorts, to onboard data, and to build the dedicated environments. And to be able to scale more efficiently, we've come up with a model that we call dynamic tenants. And this dynamic model really just shifts the paradigm as to how tenants are related to an enclave. And it merges two things together. It merges what previously we call a data use request, now data access request, with the key component that kind of defines a tenant, which is a phenotype. And so what we do here is we embed the phenotype logic directly into each data access request. And so then we allow data environments to be created automatically, and they get provisioned dynamically, and they become tailored to a specific research question and not a pre-built domain. And so imagine your data access request, you enter the typical title and abstract and justification, but then you also attach a phenotype to that, your patient inclusion criteria. So here, there's going to be a phenotype builder. You can specify all kinds of things like demographic criteria and conditions and lab results, medications, and all other criteria that are accessible through data present in N3C that can then be attached to a data access request, along with whatever linked data sets are of interest to that research problem, and then that gets submitted for approval. And what this does is it really enables rapid and reproducible research at scale without duplicating infrastructure. And it maintains institutional control and data governance fully intact. And so every project, every data access request becomes a tenant of its own. And what this also accomplishes, which is really important, is it maximizes, this is the best case in terms of the alignment of data access as precisely as possible with scientific intent. And so this is a pretty substantial shift and departure from the prior static model to something that can support really an infinite number of projects that are tailored to a variety of different research questions and clinical domains. And so naturally, the extension of that is there needs to be a community that facilitates the development of phenotypes, because now there's such a central component of each project or data access request. And so the challenge here is very clear, is that phenotype development, anyone who's done it, knows that it's very challenging. And in practice, developing phenotypes is often ad hoc, it can be inconsistent and siloed, and so it makes it very difficult to reproduce results or to build upon previous work. And so here what we wanted to do is leverage really the success of concept sets in N3C and employ that model for phenotype development. And the result of that is really a community-driven and a transparent phenotype ecosystem. All phenotypes, like concept sets, will be versioned and made available for reuse by people in the enclave. There's also going to be a dedicated phenotype sandbox where researchers can go in and build and test and refine phenotypes safely, and no data contributor approval required. And what this does is it enables researchers to very quickly, in the same way we saw with concept sets, build on each other's work. And ultimately what that does is it promotes not only consistency, but also reproducibility and the development of shared standards across the board and across the community. Another challenge is how can we ultimately balance scale with institutional control? So as the number of research projects and data contributing institutions grows, maintaining both the review and approval workflows for each project becomes resource intensive and slow. And as I previously highlighted, in the clinical pilot, we were looking at institutional control at the tenant level. And what we've developed since we received feedback is a dynamic opt-out model that gives institutions control at the level of individual data access requests or projects. And so the way that works is this, is that institutions will get access to a dashboard, and the institutions will be automatically alerted when a new data access request is submitted that includes their data. So that phenotype that investigator develops, that will ultimately be run against the data, and if an institution's data is included, they will receive an alert. And then designated institutional representatives will review those incoming data access requests in an interface, in a dashboard, and then institutions can explicitly approve, reject, or take no action. And the model is opt-out. So if no objection is raised within a defined time window, then the request is automatically approved. And the end result of this is that institutions retain per DAR control, ensuring that data is only used in projects either they implicitly or explicitly support. And so this really kind of threads the needle in terms of connecting things, scalability on the side of the scientific questions and the data access requests, all the way down to institutions' data that are being contributed to these research projects. And to go along with all of these new developments, we're going to have a refined set of agreements. Now, these agreements will explicitly state that the data contributor will have control over which data access requests will have access to their data. Data access requests will have a 1-year term, and they'll be eligible for 2 renewals. So data access requests are time-bound for a maximum of 3 years. Investigators can still continue that line of research. They will just have to submit new data access requests that will go out to the institutions providing the data for another round of clearance. It's going to have that same HIPAA limited data set that's been contributed previously in N3C, and there will still be data linkage through the PPRL mechanism that we have. And the data contribution that institutions will make to N3C will be a disease agnostic phenotype. So again, at a high level, that's what I touched on previously. That is patients with 2 visits that are at least 30 days apart. And our hope is that this new set of agreements, they're going to safeguard institutional control while also enabling scalable and secure data sharing. And ultimately, they serve as the foundation for this, the future of N3C and its research ecosystem. And so in this dynamic tenant model, refining the previous diagram I showed for the clinical pilot, here, the data contributing institutions have agency over participation, but then each of these, what were previously tenants, become individual projects or DARs. You can have multiple cancer DARs looking at different angles or with different phenotypes, asking similar or related questions. And then you have an entire ecosystem of DARs where institutions ultimately have agency over their data and participation at that level. You get the data linkages through PPRL and access to publicly available data sets through N3C. So what I wanted to do now was go through the entire data access lifecycle. So from submission to provisioning, just to really bring it all together. So you are a researcher and you submit a DAR. That DAR, data access request, incorporates a phenotype. You select your data sets, so what linked data sets you want to incorporate. You incorporate IRB documentation as necessary, and you specify the data access tier, 1 of 3, which is synthetic or limited or level 2 de-identified. That submission goes out to the DAC for review. Now, I do want to make it clear this is focused on the data access lifecycle. So what it doesn't show is the CADR aspect, which we reviewed previously. But ultimately, That submitted DAR, once it clears the institution, will go out to the NIH Data Access Committee for review. It will be evaluated on probability of technical success, policy alignment, and privacy. And then once that's approved, it will go out via that dashboard to all the institutions whose data matches the phenotype, where they can review and opt out within a defined review period. Once that review period is over, then the data assembly and integration happens. So the EHR data and other selected data sets are assembled. And then once these data are assembled, the tenant provisioning takes place. And so the phenotype matched records from the select institutions get provisioned inside of the project, including those linked data sets. And access to that project is restricted to those authorized users that are part of that data access request. And then from there, researchers can conduct their analysis and research in the enclave using the same tools and analytic assets that they know and love. We have the same publication export control, which are aggregate level exports, and they're all subject to formal review. And then on the lifecycle management component to the dynamic tenants, DURs or DARs are time limited and they're renewable twice with DAC review. Now, with all of that exciting, these exciting new changes to N3C, I wanted to also revisit the question of why should any one institution or investigator or researcher participate in N3C? And so I wanted to tell you a bit of the story. So this is imagine if, and this scenario is probably true for many of you on the call right now. Imagine if you wanted to study sepsis with 5 other institutions. What would you do if you did not have N3C? I imagine it looks something like this. You have a kickoff, you identify your partners, you initiate data sharing agreement negotiations, you start individual IRB processes, and you go through that whole administrative setup. And then 3 months later, you begin legal negotiations and you finalize your institution-specific agreements. You address any outstanding privacy or security or legal compliance issues across the multiple institutions. And hopefully by month 7, you're ready to start working on the technical infrastructure setup, where you either build or you procure a collaborative platform and you establish the compute environment or environments where that research project will take place. And then a few months after that, all the technical infrastructure is in place, and now you move on to data harmonization. This is where you have to go through and you have to clean and you harmonize data at each institution, and maybe you link external data sets, which you either purchase or you acquire via other means. And then hopefully by the end of month 12, beginning of month 13, research can begin and the project can actually start, and the data are ready for analysis after what is almost inevitably significant delays. Now, within this new model, this N3C dynamic tenant model, you have a kickoff where you submit a data access request. All data in N3C are under a single master data transfer agreement, which is already executed. And then there is that rapid institutional opt-out review process that takes place. A couple weeks later, you are ready for data and workspace provisioning. This is where that secure dynamic tenant is created. The data are already harmonized to OMOP and N3C, and the data linkages have already taken place. And so it is all around data and workspace provisioning. And really, by the end of the month, if that, you can begin your collaborative analysis in a secure and scalable environment in N3C. And really summarizing the points through that example, participating in N3C will ultimately lead to reduced data contribution burden. NCATS provides resources, guidance, and support, and you have a one-time simplified institutional agreement. It enables you to pursue research questions that are of interest to you, but also it provides you with a shared space for collaborative research projects across institutions. It reduces fragmentation. So having a centralized and scalable platform, it prevents or minimizes duplication, and it facilitates people to rapidly initiate multi-institution studies. N3C, as many of you have already experienced has advanced data cleaning and harmonization. And so we handle data cleaning, we handle standardization, and at your request, we will return the enhanced data sets to you. So if you'd like a copy of the curated OMOP payload for your institutions, we can deliver that back to you. We also have data quality reporting that we've been doing for years. So data contributors receive detailed data quality scorecards, and that ultimately guides internal improvements even at the institution itself. And N3C provides immediate access through data linkages to difficult to obtain data sets. And this is an area we're actively engaging in and working in to try to expand and provide more value to N3C users. And so ultimately, what N3C aims to do through participation of the community is to lower cost and maximize efficiency. So it dramatically reduces setup time, administrative burden, and also operational costs. And so what are the next steps? So first and foremost, we have website updates that are coming very soon. N3C is moving to this new domain, which is n3c.ncats.nih.gov. We will be sending out, so this webinar is recorded, these slides will be shared, and you will have access to the new website with updates that are coming soon. The new set of agreements that I highlighted previously are under final review, and you should expect them for execution shortly. And really, this is a call to all of you. If you are interested in helping us stand up this new model, I'm sure as we go through things, we're going to have to iron out certain kinks. We'll find bugs that we need to fix and refine operational processes. We are looking for early adopters, so please reach out. And N3C is actively working with other NIH resources like data counts and linkage across the board. And this is something Joni had touched on. And we are working diligently to relaunch the community forum on a regular basis. So we'll send out more when we're ready about that. And you should also keep an eye out for Google Forms, where we're going to ask to collect your thoughts. We're going to get feedback on notices that are relevant. So for example, there was a recent notice on protecting human genomic data in the context of generative AI. We would love your feedback on this and on more, and also to help us rejuvenate and kind of reorient and organize the domain teams that were such a critical part of N3C. And so with that, I wanted to point you to one of two email addresses at the bottom. So if you have just general inquiries, please email ncats-n3clinical@mail.nih.gov. You can also reach out to me directly at hytham-sidky@nih.gov. And with that, I wanted to kind of usher in this new era. We, in a transitionary period, we refer to N3C transitioning from the pandemic into the clinical pilots, referring to it as N3C Clinical. But now we are fully embracing this new model. And so N3C is now National Clinical Cohort Collaborative N3C. Thank you.